The healthcare and life sciences ecosystem is highly complex, comprised of different stakeholders with different goals and objectives. Patient is at the center. The other stakeholders are providers who treat the patients and prescribes medication. The pharma or biotech stakeholder develops and sells those medications, and the payer, the health insurance companies, pay for it. In the pharma and biotech industry, vast amounts of data are generated daily across the entire drug development continuum, from discovery and lead optimization to preclinical and clinical development, commercial and manufacturing stages. Despite the staggering volume of data, much of it remains proprietary and unpublished, especially data from drugs in development and failed drugs. This information, although valuable, is often inaccessible to researchers and clinicians in general. Imagine if drug discovery scientists had a tool built on a large language model that could query this pool of information and extract relevant insights in no time. Access to this diverse data and intelligence pool could revolutionize their work. While open-source large language models like a standard public large language model can provide access to the latest information, they are not trained on specialized and proprietary life sciences and medical data. Let me show you how. I am now in a public standard LLM and I'm going to ask a question. Which mutations do children carry with juvenile myelomonocytic leukemia, JMML? As you can see, it provides 3 common mutations: NF1, PTPN11, CRAS, and NRAS. This may not be sufficient for a scientist trying to design their next experiment because they want more information. We are now going to open our own tcgmcube GenAI enterprise search engine, which is our large language model, and we are going to ask the same question. In fact, we did get all those answers that the public standard LLM gave, but it has more, such as SETBP1 and JAK3. The query also returns the references where this information came from, because the local LLM model was trained on a subset of information. The references being a publication with PMID ending with 7648, provides the basic information, and another publication with PMID ending with 2011, which shows a whole exome sequencing that was done which identified these secondary mutations SETBP1 and JAK3. While the public standard LLM query did not provide this information, but it showed up in our local large language model. We have trained our local large language model on highly specialized scientific and medical information that we have been collecting over the last 2 decades. Data on protein structures, chemical structures, drug labels, clinical trials, patents, genomics, clinical data, other publicly available data. Over 3.3 million biomedical entities, 440 million relations, and 40 million documents have been mined and curated. Not only that, but global knowledge graphs have also been created using AI by creating the ontologies, contextualization, applying the semantic modeling, and fairyfication. And that's why when tcgmcube's local LLM was used, it applied the knowledge graph and the semantic search and returned all the necessary information as the query result versus the public standard LLM. Let's try another query and see what answers are generated by the public standard LLM versus our local large language model. I am now in the public standard LLM and I'm going to ask another question. Which genes are inherited and related to Alzheimer's disease? As you can see, it provides the major genes associated with Alzheimer's disease include these 6, as well as some information related to it. While this response is extensive, it may not be sufficient. I'm going to run the same question in our local large language model. You can see it has returned those listed in the public standard LLM, but has also returned more along with the references. Let's go to the references. The Genetics of Alzheimer's, which was published in 2012, it has the basic information. Also, let's go to this reference on rare coding variants associated with the Alzheimer's, published in 2017, where it talks about the rare coding variants PLCG2, AB3, and TREM2. So, this is another example how not just the main genes, but also the rare variants are captured as responses in our local large language model. And this is because of the semantic search that is applied using the ontologies. In summary, it's fair to say that open-source LLMs not just hallucinate, that is giving false positives, but also misses reporting critical information, false negatives. Missing such critical information, which we call known unknowns, can be as detrimental as hallucination. Our local LLM has been trained on our owned and specialized data while open source trained on publicly available data. Now, your proprietary data such as toxicology reports, PK reports, clinical trial protocols, regulatory reports, and other proprietary documents for successful, ongoing and failed molecules can now be used to train the local large language model to provide faster and higher accuracy in responses and bring in more productivity to your enterprise and industry search with high velocity. Our application pinpoints the specific articles taken as references, whereas the public standard LLM provides references at an institution level.